to the next session, which is tumor science, apoptosis, and cancer. I'm going to begin by talking about the activity of a thyroid hormone derivative, tetrac, as a nanoparticle, its activity against glioblastoma models. Many of you know glioblastoma is the commonest malignant brain tumor. The median time to recurrence after standard therapy, and standard therapy is surgery, radiation, and chemo, for example, with Temodar or temozolomide, is only 6.9 months. The 5-year survival rate with standard treatment is 8%. GBM cells, like most malignant cells, generously express plasma membrane integrin alpha V beta 3, protein I mentioned earlier. The extensive blood vessel supply to the tumor also expresses the integrin. Alpha V beta 3 contains a specific cell surface receptor for thyroid hormone, as I mentioned earlier, and for the thyroid hormone derivative, tetraiodothioacetic acid, tetrac. You saw the diagram earlier on the left portion of this slide, the cartoon of the integrin. And the X-ray crystallographic analysis of the integrin has revealed the different angles at which thyroid hormone analogs, in fact, fit into this site. And the purple site here, the tetrac, fits nearly across this groove between the alpha V and the beta 3 analogs and explains its ability to block T4 and T3 binding. But in addition, in the absence of T4 and T3, tetrac has its own life as a modulator of gene transcription and of the crosstalk between the integrin and nearby vascular growth factor receptors. The nanoparticulate formulation of tetrac is nanotetrac, and it's limited in action to the integrin. We put the PLGA nanoparticle covalently on the tetrac, so that in fact the tetrac, a small molecule, can no longer enter the cell. And the reason we did that was that unmodified tetrac gains access to the cell interior where it's a weak thyromimetic, in other words, just like T4 and T3, and expresses unfavorable, particularly cardiac side effects. So we're functioning primarily on the outside of the cell. From the integrin, nanotetrac downregulates expression of multiple cancer cell survival genes, and these include EFGR. It promotes apoptosis by multiple pathways. It blocks anti-apoptosis defense. It blocks actions of multiple growth factor receptors, and it upregulates expression of thrombospondin 1. So it has a panel of remarkable effects that are primarily expressed in rapidly dividing blood vessel cells and tumor cells. Here is the diagram of tetrac. As I said earlier, it's missing a carbon and an amine group in the side chain. And what we've done via a linker is connect it to the PLGA nanoparticle. There are 5 to 8 of these tetrac linker combinations covalently bound to them that compose the nanoparticle. And the nanoparticle is about 150 to 200 microns in nanometers, in diameter. Now, I said a number of genes were affected by nanotetrac, and here you see in green increased gene transcription, which is desirable clinically. For example, caspases, ChIBI1, inhibitor of the catenin pathway, P53, P21 are all upregulated, and of course BCLX, short form. Decreased gene transcription, also desirable. Nanotetrac downregulates EGFR, it downregulates the X-linked inhibitor of apoptosis, it downregulates NF-kappa B, and downregulates 8 of 9 cyclins, and also cyclin-dependent kinase. Now, because alpha V beta 3 is generously expressed on tumor cells and on rapidly dividing endothelial cells, these effects are relatively limited. So we've seen almost no side effects with this drug when it's administered to healthy animals. The alpha V beta 3 simply isn't expressed very widely. The present studies examine the susceptibility of human glioblastoma U87 cells and C6 rat glioma cells to the actions of nanotetrac. And we use 3 different systems. Rat C6 glioma cells were engrafted into nude mice. And the mice received daily combination suboptimal nanotetrac, 0.3 milligrams per kilogram, and standard dose Temodar. We wanted to see whether or not Temodar or temozolomide, standard therapy for GBM, was improved in its potency with a suboptimal dose of nanotetrac. We used U87Mg luciferase glioblastoma cells in other grafts, and here we looked at a single intragraft injection of either unmodified tetrac or nanotetrac up to 10 micrograms. And we followed the animals for 16 days. 1 dose administered at the time of the implant. And the third model was U87Mg cells engrafted in nude mice. The animals were then treated daily with optimal doses of nanotetrac, 1 milligram per kilogram times 10 days. There are some other background information here. We measured xenograft volume, we measured tumor weight, and we measured hemoglobin as an index of vascularization, and we did blinded histopathology and necrosis assessments. We also did some IVUS scans of the U87Mg luciferase cells. So suboptimal nanotetrac dosing in conjunction with Temodar. What you can see here is that Temodar was not very effective at its optimal dose. In fact, it had no effect on growth of cells. Suboptimal nanotetrac also had an insignificant effect versus control. But if we added the two together, suboptimal nanotetrac and temozolomide, we actually saw a temozolomide effect. If we looked at this histologically and graded via a necrosis index what was happening in the cell slides. In the tumor slides, you see the controls were vigorous, temozolomide also vigorous. With nanotetrac, well, there was a little bit of necrosis, but not impressive. With nanotetrac and temozolomide, we saw pycnosis of nuclei, and we also saw apoptosis with membrane lysis going on in many of the cells. We then looked at U87Mg luciferase xenografts inoculated with a single dose of nanotetrac. And here we see that tumor weight dramatically affected by tetrac at a high dose, by nanotetrac at a low dose, and by nanotetrac at a high dose. So all 3 of these approaches were effective. As I've said, unmodified tetrac has some undesirable side effects, and so it is not a valuable alternative, a useful alternative to nanotetrac. Nanotetrac also has some extended gene expression effects which are desirable. If we looked at the IVUS scans of the control versus the void PLGA, and using the scale where red is entirely viable cells and blue or black are dead cells, you can see that the control cells flourished. The void PLGA had no effect. Low-dose tetrac, high-dose tetrac had some effect, low-dose relatively little effect. Nanotetrac at a low dose was pretty effective. At high dose, 10 micrograms per transplant, there was essentially no viable cells on IVUS scanning. And if we looked at the hemoglobin content of the tumors, reduced by 50% or more by all three drugs. And we've shown this in a variety of tumors, that tetrac and nanotetrac are effective anti-angiogenic agents. The third model was the daily treatment of U87Mg xenografts. And here you can see the controls did well. The nanotetrac treated animals treated at an optimal dose, 1 milligram per kilogram. The weight plummeted, decreased by almost 50%. And if we looked at the histopathology, the tumor was healthy. There was just about complete necrosis in the tumor after 10 days of treatment. Here's the necrosis index, multiple slides looked at blindly by the histopathologist. And again, what we saw here was an extensive mass of dead cells, pycnotic cells with pycnotic nuclei. Some living, we saw a few cells that were still dividing, by the way, and nanotetrac stops DNA, stops double-strand DNA break repair. So, in fact, dividing cells are highly susceptible to nanotetrac therapy. So we were pleased with the extent of necrosis and apoptosis that was induced. In summary, in 3 xenograft model systems, relevant to glioblastoma multiforme, nanotetrac reduced tumor volume and weight, widely induced necrosis and apoptosis. Suboptimal dose nanotetrac potentiated the antitumor effect of conventional dose Temodar. Unmodified tetrac has anti-tumor activity but has reduced systemic potency compared to nanotetrac, has a limited effect on cell survival pathway gene expression. We've just published this information, and has an unfavorable systemic side effect profile. So it's not a drug of choice for us. I just want to show you in an overview in 2 tables of the transcriptional mechanisms by nanotetrac, by which nanotetrac expresses its presence. In terms of angiogenesis, it decreases BGF gene expression, VEGF-A, EGFR transcription, TSP transcription is increased on the other hand, MIR21, which is oncogenic, is decreased, MIR15A, which is pro-apoptotic, is increased. It's a favorable side effect profile, certainly, for the drug in terms of angiogenesis. And if we look at the cancer chemotherapeutic actions of nanotetrac, we have a variety of effects. For example, there's decreased efflux of doxorubicin, of etoposide, of cisplatin, and of etoposide, and that's at least in the case of 2 of those drugs, is a PGP effect of the drug, but cisplatin, that effect, it's not a substrate of PGP. So how we are increasing residence time of cisplatin is not yet clear. There's radiosensitization. These data have been published. There's disabling of the cell survival pathways in a variety of ways. Caspase is affected. The BCL2 family affected. The catenin pathway, matrix metalloproteinase is affected as well. And then there's the cell cycle effects. So in conclusion, directed at the thyroid hormone tetrac receptor on alpha V beta 3, which is widely expressed in tumor cells and in blood vessel cells, systemic and local nanotetrac is capable of inducing widespread necrosis in xenograft models. Nanotetrac induces cancer cell apoptosis, inhibits anti-apoptotic mechanisms of defense, and it's anti-angiogenic. The agent to date has not been shown to be cytotoxic, even in relatively high doses. Questions? Yes, Steve?
In your xenograft models, does nanotetrac shrink the tumor to nothing, or does it plateau?
Yeah, it depends on the cell, on the tumor cell line that's used. In the case of U87, it decreases by 50% and stabilizes. And if you look at the histopathology of that, it's detritus, basically, that's there. In some of the other cell models that we've used, there's rapid cleanup, and so the tumor continues to decrease. So tumor in quotation marks continues to decrease there. So it's really is how the host is handling the dead cells that determines what the endpoint will be in terms of tumor size. Yes.
There you go, sir.
Did you see any difference between 2D culture and 3D culture vis-a-vis the effect of this substance?
No. We've reproduced all this began in vitro, And we've reproduced, I think, all of the findings in vivo.
Do we have any other questions? Dr. Davis? Very good.
Sorry, I have one more question.
Yeah.
I'm just wondering, you mentioned a cardiotoxic effect after internalization. I'm wondering, because macrophages express alpha V beta 3, and is it possible that part of your tumor reduction is due to, and it would be consistent with the slower clearance of apoptotic or cell debris as well?
It's a terrific question.
If it's cytotoxic to monocytes.
Yeah, absolutely. So platelets and white blood cells, macrophages, all express alpha V beta 3, and so there is concern that in fact we might impair the cleanup. But nonetheless, what we've seen is cell death. The cleanup may be delayed. I don't have good evidence for that, but it needs to be that issue needs to be explored. But the cell death is so widespread that I'm pleased with the results. But measuring tumor volume, for example, as an endpoint, it could be you could see less of an effect than you might optimally want. That's an important issue. Thyroid hormone is so important to macrophages. Regular thyroid hormone, T4 and T3, to clean up bacteremias. A recent paper by Chen looking at meningococcemia clearance by a nitric oxide dependent process. It's thyroid hormone dependent via the integrin. And obviously, if you have an infected tumor patient, you don't want nanotetrac on board at that particular time. Yes?
How quick is the cleanup?
We've only done studies to 28 days, and it stabilizes, I would say, by 15 to 20 days. That's it. And we don't see much more after that. But in the first 2 weeks, we see, I don't have the slides with me, but actually in the first 5 to 7 days, you see a dramatic decline in tumor size. That is two functions. One is there's some cell death, but you still have some fluid there that has to be cleaned up. But the vascularity has been dramatically decreased. When we've looked at this in the chick chorioallantoic membrane, for example, putting human non-small cell lung carcinoma into the chick chorioallantoic membrane, letting it grow and then treat it, and it vascularizes beautifully. The first thing that happens is there is systematic loss of the vascularity of the tumor. Without hemorrhage, you just see very nice breakdown with some macrophages there cleaning up the vascularity. So loss of the vascular supply explains some of that volume loss.